So let's start today's seminar. Today we have Dr. Tristan Bepler. He will be talking about the very exciting recent development of language model, protein language model. So Dr. Bepler is a group leader currently of the Simon Machine Learning Center at New York Structural Biology Center. And he is also a co-founder and CEO of Open Protein AI. So he's received his PhD from MIT in computational systems biology program. And his supervisor was Professor Bonnie Barga. She is a very well-known figure in computational biology. She did lots of groundbreaking works, including this protein language model recently. And she is also recently working on cryo-EM. Then after getting PhD, he also did a postdoc with Tim Liu at MIT. And then he joined at the structural biology, Simon Machine Learning Center at New York. But interestingly, he is located currently in Singapore. So I really appreciate that he's staying up late and then giving us the exciting lecture here. Tristan, please.
Thanks for the nice introduction, and thanks for inviting me to come talk. So today, what I'm planning to talk about is learning from evolution to design new proteins. So this is a lot about protein language models and how we can use those to learn sequence function relationships, and also use those for protein design, in addition to actually making predictions about proteins from their sequences. As mentioned, I am a group leader at New York Structural Biology Center. So I also do a lot of work on protein structural biology and develop machine learning methods for analyzing cryo-EM data and protein structures in cryo-EM, especially a lot of geometric deep learning algorithm and software development there. So that's not what I'm going to talk about today then. So today I'm going to talk more about work that's been going on now at openprotein.ai, but it started when I was at MIT. To give you a brief outline of what I'm going to talk about, I'll start with some introduction, some really brief background on proteins and evolution. And I'll talk a little bit about some older school kind of generative models of protein families. And we can think about sort of the relationship here with classical statistical sequence modeling and evolutionary based variant effect prediction. Then I'll go into large protein language models and briefly discuss some of their benefits and limitations, which will motivate some of our newer work in the space. So the Protein Evolutionary Transformer with POET, which is a generative model of protein families as sequences of sequences. And hopefully that will make sense when I get there. And then we'll move on to moving beyond evolution. So thinking about transfer learning for quantitative sequence of function prediction and protein optimization, and then we'll have some conclusions and future directions. Also to mention, I hope to leave some time for questions. Wasn't sure exactly what the format was in terms of how whether I would be interrupted during the talk or not. But happy to be interrupted if that's how you guys do things. Otherwise, there should be time for questions at the end as well. So proteins are the workhorse really of the molecular level of life. They are polymer chains, these sequences of amino acids. That sequence is encoded in genes. So DNA, the DNA in genes encodes the sequence of amino acids. And those chains fold into three-dimensional structures based on physical interactions between those amino acids. And proteins themselves then carry out the vast array of molecular functions. So they catalyze reactions, they form cellular or viral structures, they bind to each other and to membranes and to small molecules. They transport molecules. They replicate DNA. Basically, most of the sort of function at the molecular level of life is carried out by proteins. So understanding this sequence to structure to function relationship is really critical, because ultimately the function of proteins are determined by their sequence of amino acids. And so that's really important for understanding the functions of new proteins, designing proteins for industrial therapeutic uses. Proteins are widely used in industry for things like biosynthesis of small molecules, chemical engineering, bioremediation. They're also widely used as therapeutics. Antibodies are proteins. And also for understanding actually disease-causing mutations, right? And diseases, especially like cancer or genetic disorders, oftentimes, there are what's our so-called like missense mutations, which actually are mutations in the DNA that cause a change in the amino acid in a protein and therefore produce a pathogenic effect. So it's important to be able to understand, when those happen and be able to understand what happens to the protein so that we might be able to, say, drug that and actually cure someone. So proteins are these things that exist in nature. They're encoded in genes. That means that they are subject to evolutionary pressures. We have massive databases now of protein sequences that we know from existing in nature. So with the rise of next generation sequencing, people are going out and sequencing every genome they can get their hands on. And then as a result, we know the sequences of the proteins that are encoded by those genomes, and they wind up in databases like UniProt. And evolution itself fundamentally is a Markov process. So over time, mutations are proposed. In other words, mutations that occur in DNA that could cause a change in a protein, and then they're selected. If that mutant is fit, the organism survives and reproduces, and so we see more of that protein, we see more of that mutation. If that mutation breaks the protein, has a fitness cost, then the organism dies or fails to reproduce, and then so that mutation is selected out of the population. And so as a result of this, evolutionary process over billions of years, we now have the vast array of and diversity of proteins that exist in nature. And so all of those proteins are constantly under selection for being functional. And ultimately, because that functionality is important for the organism to be fit. And so that means that evolution is basically selecting for stable and expressible and functional proteins. And so there's potentially a lot that we can learn as a result just from looking at natural protein sequences. So statistical sequence models are basically a way to actually try to learn functional constraints on protein sequences from this kind of evolutionary data. So a really classic example of a statistical sequence model would be something like site-wise factors models. This could be like a position-specific scoring matrix, where in essence, we take a protein family and we calculate a multiple sequence alignment of that family, so we know, okay, how the amino acids match up between those proteins at each position. And you can do something really simple, like just count, okay, at each position, how often does each amino acid occur? That's a very simple generative model of a protein sequence. You can actually generate, it's generative because it models that distribution. You could also actually sample a protein sequence from it by sampling the amino acid at each site independently. You can calculate the probability of a sequence under that model by taking some new sequence in that alignment and calculating, right, what's the probability of each amino acid at each site, and the product of those would be the probability of the whole sequence. So that's a very simple generative model of protein family. We can have more complicated models that include things like pairwise factors. So an example of this would be, say, POTS models, which is like a version of an Ising model, where you have these basically second order terms where for every pair of positions, we're actually learning the joint distribution over those amino acids and estimating them from multiple sequence alignment. And there are newer methods like neural network-based methods where maybe you have some latent factors that you actually then describe it with a neural network of how you can actually generate a sequence from this latent factor in something like a variational autoencoder. And so we can use these kinds of models to calculate or to estimate like what the fitness of a mutation might be under this evolutionary model. Because in essence, you could ask, okay, for some mutation, I can calculate the probability of that sequence. I can also calculate the probability of a wild type sequence. And we could say, look at that like log likelihood ratio to get an idea of whether this mutation might be fit or not fit. Because if that mutation is much less likely than the wild type sequence, then we think there's probably some fitness cost to that. We don't observe that mutation in nature, or we don't think we're likely to observe that mutation in nature. Whereas if it's a roughly equal likelihood or even higher likelihood, then we think actually maybe this mutation would actually confer fitness advantage. And so neural network models of protein families are actually highly predictive of variant effects. So if you fit, say, like one of these neural network models on a large protein family, and then you took that model and you were to calculate the likelihood of, say, every amino acid substitution in that protein, and you were to compare that with actual functional data collected in, say, like a deep mutational scanning experiment, you would find that, generally speaking, there's a strong correlation. So that likelihood of the sequence under these kinds of generative family models actually correlates with whether that sequence is actually functional in a lab assay. But there are definitely limitations to these kinds of family-based models. So primarily, they're limited by available homology information. So we could actually think about, especially for simple models, how much data you would need to actually be able to reliably estimate the parameters of that model. So if we're making a very strong simplifying assumption like that, like that of a site-dependent model, or PSSM, well, to estimate those parameters reliably, you need enough sequences to actually cover variation relatively deeply at every site in your protein. So that already requires a pretty large number of homologs. If we then think about going towards something like a pairwise couplings model, now you have something on the order of the length squared times like 400 parameters in that model. So even for modestly linked proteins, actually the number of parameters in that model becomes explosive. And we can, similarly for neural network-based models, right? If you want to scale that model up, get more predictive power, you need to have very large families. You have to know the sequences of an enormous number of homologs. And the primary reason for that is that these models really can't learn any kind of general protein properties. You train the model on one family, you fit it to one family, and that's it. The model doesn't benefit from any information that you could glean from other proteins out there in nature that aren't from this family. They have to be trained on family by family basis. So, this sort of motivated some of the early work in protein language models themselves, like larger scale protein language models, because ultimately the question was, well, how can we actually capture information across evolutionary scale, right? How can we learn from all protein families in order to be able to better make predictions, especially for smaller protein families, orphan proteins, where we don't have tens to hundreds of thousands of homologs that are known. So databases like UniProt contains hundreds of millions of unique protein sequences, tens of millions of homology distinct sort of clusters of sequences. And so we can use that data to actually train neural networks to capture this information across the whole protein universe. And there's a couple of ways that we can do this. So basically, we can think about purely generative models, things like autoregressive language models. So in this case, we would be doing something like basically making doing a autoregressive factorization of a protein sequence, similar to something like a profile HMM, where what the model actually is doing is it says, okay, given the first I amino acids in a sequence, I want to train this model to predict what is the I+1th amino acid. And basically what that means is you could apply that model autoregressively via like the probability training rule to calculate the probability of a whole sequence. And the other really nice thing about this kind of generative model with protein sequence is you can actually draw samples from it and calculate exact difference. But these models do have a limitation, which is that if you're interested in thinking about, well, like, what is the model learning about a specific site in this protein? Well, that's only conditioned on the amino acids that come before it. It's not including any information about sort of all of the amino acids in the sequence. And so that's motivated two different approaches. So an earlier approach to solving this problem was thinking about something like bidirectional language models, where the concept is basically we have one model that goes in the forward direction, and we have another model that goes in the reverse direction. And so then to understand what's at some site or have a representation of that site that sort of is conditioned on the whole sequence, we just take some representations from each of those models. But then even more recently, coming out of the ideas in NLP, like BERT, is the idea of using actually a masked language model. So in this concept, what we would say is, okay, I'm actually going to feed this whole sequence into my model, and my model is fully connected in the sense that it can use every position to make predictions about every other position. And so in order to prevent it from just sort of trivially memorizing things because it sees a G and it can output a G, Well, what we'll do is we will randomly mask out positions and we'll train the model to recover it. In NLP, this is sometimes called a close task. It's like a mass infilling problem, where in essence, it's saying like, if I removed a word from a sentence, but you got to read the rest of the sentence, you can usually take a pretty good guess at what are possible words that could go there. And this is the analogous problem for a protein sequence. So in essence, what all of these kind of rely on is the idea that, well, if you don't get to know what this amino acid is, but you could guess what it is, that means you must understand this protein, right? If you can look at this protein sequence context, except for some amino acid, and say reliably, what is that amino acid? What must that amino acid be? Then that model must understand this protein sequence. It understands something about the sequence constraints, which comes from functional and structural constraints. Okay, so it turns out actually these do work very well. Large protein language models do learn vector representations of protein sequences that are predictive of protein structure and function. That information is in the sequence, and these models do learn it. So things like secondary structure prediction, we could benefit tremendously from using representations from protein language models. We can examine the internal states of protein language models to actually be able to extract evolutionary statistical couplings, which are highly predictive of things like residue-residue contacts within protein sequences. As an aside, we can also sort of bake some of this information into these models when we train them. We don't have to only use sequence modeling tasks. We could say also train a model to simultaneously do something like predict structural properties or functional properties. But really, ultimately, the big thing that these models rely on is scale. So number of parameters. This is something people talk a lot about in natural language processing, and essentially all the same concepts end up applying for proteins. Because we have so much sequence data available, you can basically continue to add more and more and more parameters to these models, increase their capacity, and they get better and better and better and better at understanding that the whole sort of evolutionary landscape of proteins, and actually also end up getting better at understanding sort of or being predictive of the relationship then between, say, like sequence and structure. So today, the largest protein language model that I'm aware of is Extremo, which has 100 billion parameters. So to sort of put that into context, this model would require, like, you can't even fit this model on a single state-of-the-art GPU. You can't even fit the parameters of the model. So let alone use the model. Right. So the question is why? Like, why do we need to scale these things to be so massive? Well, it's really because this version of a protein language model has to memorize the entire protein universe. It doesn't understand, it doesn't get to see evolution. It doesn't get to understand anything about, say, like a set of the proteins that are specifically a member of a family. In order for this model to be able to make predictions, it has to actually bake into the parameters of the model everything that's in UniProt. So you need a lot of parameters. Same problem in NLP, right? Models like ChatGPT are also billions of parameters now. I think actually in NLP, people are also at the like 100 billion parameter, probably slightly beyond this scale. And it's because these models, you have to just memorize the entire data set. They have to memorize the entire corpus store. So this scaling requires immense GPU compute resources for training and inference, and that's time and cost prohibitive. So one of the fundamental questions we wanted to ask is, are there ways that we can actually overcome these scaling limitations? How can we make protein language models more efficient so that we don't only get performance improvements from just doubling or multiplying the number of parameters by 10, which doesn't really, is extremely impractical, especially given amounts of compute we have available. So we really need to make these models more efficient, more parameter efficient, and that's I think a really fundamental problem in the field. So one idea we thought about was basically, how can we actually leverage homology information directly in a protein language model? So the kinds of language models we were just talking about essentially just look at a single protein sequence. So all homology information has to be baked into the parameters. Can we not require that to be baked into the parameters. So what we worked on is a model which we are calling the Protein Evolutionary Transformer, or POET. So this is a generative model of protein families as sequences of sequences. So let me explain sort of what that means. Basically, what that means is rather than this model only generating one protein sequence, it actually generates many protein sequences, auto-regressively in order. So what that means is we're actually going to model the probability of an entire protein family. So let's say you have a protein family within sequences. The probability of observing that family, that whole family, is basically the probability of observing all of those sequences. And we can factorize that in sort of a similar way that we can factorize the probability of a single sequence in terms of its tokens. We can factorize the probability of a family in terms of its sequences. So we can say, what's the probability of seeing sequence I, given that we've seen all the sequences before I? And then within that, we can model the probability of a sequence as being, well, what's the probability of seeing sort of the amino acid at position J, given all of the positions before that in this sequence, but also all of the other sequences we've seen so far in our family. So in essence, this like transforms this problem from just the problem of autoregressively modeling one protein into the problem of autoregressively modeling many protein sequences. So this introduces some problems, but essentially that's the idea. So in order to make this really work for protein sequences, especially where we're thinking about now, we have this like set of proteins, we have this bag of proteins, and we shouldn't really care too much about how they're ordered, right? Like the probability of the sequence I shouldn't really be sensitive to the specific ordering of the sequences that came before it in the family, because that doesn't really have any intrinsic meaning. So I had to come up with a specific sort of attention architecture, which I'll describe in a second, in order to make this work. And we trained this on hundreds of millions of sequences and UniRef 50 clusters. So now one training data point is essentially one protein family, which we're viewing as like one UniRef 50 cluster. So let me dive a little bit into this attention mechanism. So what's really important here is the ability to scale to long context lengths, because now we're talking about a sequence of sequences. So one protein sequence might be a few hundred amino acids, 1,000 amino acids would be a very long protein. But now if we're talking about a family, we're talking about tens, hundreds, thousands of protein sequences. So that context length of that sequence becomes very long. And we also have the fact, this other factor, which is that, we care about conditioning on previous sequences. So basically to generate like the ith amino acid in my current sequence, I should look at the amino acids that came before it in this sequence, but I should also look at all of the sequences that I've seen before. And there isn't really any positional information that's relevant for those sequences because we don't care about the ordering. And there is some positional information that's relevant though within the amino acids of those sequences because there's an alignment. But the other thing we wanted to do, which is important, is we didn't want to actually assume we knew the alignment. So we didn't want to feed in a multiple sequence alignment because there are problems that arise in terms of having very gappy sequences. Multiple sequence alignments can be wrong. And so we wanted to make this work for sequences that are unaligned. So we'll let the model learn the alignment. So to give you a sense of the challenge here, basically to condition on 100 homologs of length 250, which is modestly length protein, you already require a context length of 25,000 tokens. And so for reference, something like ESM1B only conditions on at most roughly 1,000 tokens. So we have, so even for a modestly sized family, we have to go well beyond this. In natural language processing, even for very large language models, the upper limit of context length currently is around 8,000 tokens. And so to scale beyond this, we really, what we're leaning on is this idea of modeling sequences as these unordered sets with this tiered attention, where we attend to previous sequences separately from attending to tokens within our current sequence. And so as a result, we actually were able to get this model to be able to generalize the context lengths of 60,000. So let me show you a little bit of data. So basically, what this is showing is a comparison between the POET transformer and a regular transformer. Looking at perplexity. So perplexity lower is better. This is our ability to predict what amino acid comes next in a sequence, where we're looking at different context lengths. So the other thing that we should observe is that as we see more homologs, our ability to model future sequences should get better, because we have more and more and more homology information to use. So we should get better and better and better and better at predicting what the amino acids are in future sequences, because the model has access to more and more and more of that information. So if we train a regular transformer with different training context links, we actually went up to 32,000 tokens during training in the context link of this transformer. What happens is the model works up to the context link that was trained on, but as soon as you ask the model to go beyond that context link, it completely fails, which results in this massive spike in error. So basically anything beyond 8,000 tokens for the regular transformer trained on 8,000 tokens, it's unable to predict. But by actually using this hierarchical attention structure, we were able to get POET to actually generalize beyond its training link. Even POET trained on 8,000 tokens, it's still improving as we show it 64,000, up to 62,000 tokens here, roughly. So POET actually is able to generalize well, despite being having to be trained on a limited context link. It benefits from seeing more sequences. Okay, so that's great, right? So this is a generative model of a whole protein family, but you might ask, well, when do I actually want to generate a whole protein family, right? I want to actually condition on those proteins, and we can. So one of the benefits of this kind of autoregressive conditional structure is that we can actually condition on homologs. We can show those to the model and use those to then condition on those actually generate new proteins or calculate the probability distribution over a new protein sequence based on our factorization. So we can actually condition POET on a specific protein family by calculating that sequence probability condition of homologs. So we can essentially use this as like a retrieval augmented protein language model. So now we don't have to ask POET to actually memorize every protein family. We can show it examples. So actually all POET has to learn is how to do inference on an evolutionary model. And it can learn across protein families how to do that very efficiently, and how to learn sort of like general protein priors to do that inference. So let's say we wanted to make some predictions about the probabilities of a variant of a sequence. Right? So we have a wild type sequence and we want to score variants of that sequence, right? Will they be fit or not? So what we can do is we can take that wild type sequence, we can go to a sequence database like UniProt, search up homologs. We can take all of those homologs, we can feed them into POET as our prompt, our conditioning. Then we can enumerate variants of our wild type sequence and calculate their probabilities conditioned on those homologs so that we get these likelihoods. Basically, we're calculating the probability of the wild type and each variant sequence conditioned on the prompt. And then we can use that to do the same thing as any kind of family-based generative model. We can calculate those log likelihood ratios and see whether they correlate, use them as fitness prediction and see whether they actually correlate with assay data. Okay, so it turns out that actually it does correlate. So this is actually state of the art for de novo variant effect prediction. So what we looked at here is we are, as I mentioned, we're calculating the log probability of the variant sequence conditioned on natural homologs, and we're comparing variants by log probability. If we go and look at a large data set, large set of mutational scanning data sets where people have actually enumerated basically like most of the single substitution variants or even higher order variants for some of these proteins, and then gone and actually functionally characterized all of those variants. There's a data set of these called Protein Gym, which contains about 90, just over 90 of these mutational scanning data sets, which is a nice benchmark data set for this kind of variant effect prediction. So when we go and look at that, what we can see is that POET actually outperforms other protein language models and evolutionary sequence models. And what's really nice here is that we also generalize across MSA depths. So something you might ask is, okay, if you have, to sort of understand this better, if you have a very deep MSA, you have a lot of homologs, you expect the model to perform better. And so oftentimes we're really interested in what happens in this sort of like low homology regime where there aren't that many homologs. And so this is where you really benefit a lot from this kind of, or think you might benefit a lot from this kind of evolutionary prior learning across protein families. And so POET really performs very well actually across the board, and performs well in the low homology regime. The other nice thing about POET is that because it's autoregressive, we can calculate the probabilities of indels. not limited to substitution variants. So you can calculate the probability of a sequence where you delete an amino acid, insert an amino acid. And so this actually makes POET very useful for not just missense mutations, but also intel mutations. Okay. The other thing that's cool about this kind of structure is that we can do prompt tuning. So anyone who's familiar with ChatGPT probably has some idea of prompt tuning. Basically, right, it's the idea that you want to optimize this like text that you feed to the model in order to get the model to do what you want. So in essence, we could do the same thing with POET, where prompt tuning involves basically customizing the set of sequences that we feed to POET to get POET to generate what we want by, because it's going to model that conditional distribution. And in particular, what it means is we can customize the homology level at which POET operates. So what I'm showing you here is some data from a paper by Russ et al., where they were doing mutagenesis of coarse meat mutase. And they were actually functionally characterizing these coarse meat mutase variants in E. coli. So what I'm showing here is basically, if we just do sort of a regular homology conditioning where we take the E. coli coarse meat mutase, we go and find all the homologs of that, we use those as the prompt for POET, and then we look at, okay, for variant sequences designed by Russ et al., how well does the POET likelihood actually correlate with their ground truth functional measurement? And we get a reasonable correlation. But it turns out that a lot of natural proteins actually don't work in E. coli, even though they're coarse meat mutases. So that means there are natural, presumably functional in their host organism, coarse meat mutases that did not work in Russ et al.'s assay. So that means our family is too broad. So what we could do is we can say, okay, let's focus our prompt only on natural proteins that actually work in our system. This is like a subfamily in essence. And if we do that, we get an improvement in the correlation, right? The model is now more predictive of whether a variant will actually work in our system because we focused it on sort of that relevant subspace. So this is a cool thing that we can do with POET. Also, it's actually a generative model. That means you can draw samples from the model, which enables new sequences to be sample efficient. So coarse meat mutase, I'm just showing here sort of a UMAP in the background of some of these natural sequences and the functional subspace, and then some sequences we actually sampled from POET overlaid. So you can see that they really do, it follows this sort of functional subspace of coarse meat mutase sequences. And if we actually take some of these sequences and go run them through a structure predictor, what we get is, even sequences with very low sequence identity to any natural coarse meat mutase, we still get that this will, it's predicted to fold into a coarse meat mutase fold with high confidence. We can do this for other proteins. We can, we could say focus POET on lysozymes and generate lysozymes. And actually, it turns out when we tried this, that we were able to generate lysozyme-like proteins with less sequence identity to any natural lysozyme, but that were predicted to fold with higher structural similarity to lysozymes than were generated by ProGen. So this is another unconditional protein language model that was actually fine-tuned on lysozymes specifically for this problem. Without any fine-tuning, you just change the homologs you're conditioning POET on, and it's a nice generative model of lysozymes. We can also use this to actually score insertions and deletions, right? So we can actually enumerate, say, every deletion of a protein and calculate its likelihood and get an idea of where in a protein deletions might be tolerated. We can look at whole fragments, right? So we could delete we could delete a whole fragment of a protein and see where fragment deletions might be tolerated. Perhaps unsurprisingly, in this protein, we think it would be pretty tolerant of deletions of the C-terminus. We can also look at insertions. So we can look at things like we can scan over every position that we could, for example, insert a nuclear localization signal peptide into Cas9. And we can calculate the likelihood of all of those variants to get an idea of where might be most tolerated to insert this nuclear localization signal. So actually, interestingly, what we find here is that the site here around position 700 something actually seems to be the best site to introduce this nuclear localization signal, as opposed to the inner C-terminus, which are where people usually put these. And if you actually go and look at the crystal structure of this Cas9 protein, because it exists, turns out this is a exposed loop of the protein. So this is a plausible nuclear localization insertion site. We can also think about, well, places we could split or circularize a protein via tolerance of a just sort of linker insertion. So here what I'm showing is now a scan where we're looking at this GGS, times 3 linker insertion. And what I've plotted as well on here are known split sites for generating circularized Cas9s. So a circularized protein essentially means that we take the original N and C terminus of the protein, we join them with a linker, and we split the protein at some other site, which becomes the new N and C terminus. So we essentially like roll the protein sequence and we create a new N and C terminus. And that can be very useful for changing sort of sites that you might want to, for modifying proteins to either sometimes make them more stable actually, but also basically making it so that you have a new N and C terminus where you could attach some other protein domain or do some additional engineering. And so it turns out that actually these sites that are tolerated as circularization sites actually come up as being pretty highly scored as being tolerant of linker insertions. So we're able to do things like reasonably predict things like where we might actually be able to split a protein and still yield functional pieces or circularize it. Okay, so POET is a retrieval augmented protein language model. It's a generative model of protein sequences or protein families as sequences of sequences. They can be conditioned on homologs to model protein fitness landscapes and generate new proteins. It generalizes to large context lengths, and it's a state-of-the-art variant effect predictor. So we actually, if you're interested in learning more about this, we have a preprint on arXiv. The paper is coming out in NeurIPS in December. And we also have web API-based access to the model through openprotein.ai, which if you're interested in trying it out that way, it's available right now. REST API, Python client, also GUI access. You could fill out the early access form if you want to try it. We'll also be releasing code for the model next week, actually. So that will also be available soon on GitHub. Okay, so that's great. Now, evolutionary modeling is very powerful, it's very cool, but it also has its limitations, right? So moving beyond evolution by learning from assay data. So evolution is this very powerful source of information. Log likelihoods are highly predictive of protein properties, but what they're actually predicting, right, is fitness. And so it just turns out that fitness correlates with biophysical properties of proteins that we care about. So fitness correlates with activity because a protein needs to have activity to be fit, usually. Fitness correlates with stability because, generally speaking, proteins that are need to be stable to be fit. It correlates with expressibility because an organism needs to be able to make a protein for that protein to be fit. Pathogenicity, et cetera. It correlates with all of those. However, evolutionary sequence models can't actually predict any of those specific properties. It can't predict stability because it doesn't know anything about biophysics. It's predicting fitness. We're learning these statistical distributions from evolution. So that also means it's restricted to what evolution is selecting for. If the evolutionary pressures on a protein are for it to have some very specific activity and not some other activity. So for example, let's say we're talking about an enzyme and has activity on one substrate, right? Maybe there's an evolutionary cost to that enzyme being very promiscuous and having activity on other substrates. But that might be something you actually want that protein to do if you want to use it for industrial purposes. But evolution doesn't select for that, right? So the question is, how can we now actually predict specific properties and be able to use those to engineer proteins, right? How can we understand sort of those specific sequence to function relationships from data? And I really want to motivate this from the perspective of protein engineering itself. So I would describe protein engineering really as like a needle in a haystack problem, right? So the actual space, the feasible design space of proteins is unbelievably massive. It's literally astronomically large, right? If we're just talking about 10 amino acid long protein or 10 sites in a protein, right? There are 10 trillion possible sequences, right? Because there's 20 amino acids that could go at each site. So there's 10 trillion possible sequences. Even if we were going to use something like a reduced alphabet like 13 amino acids, you still have 100 billion possibilities. I think if you have a sequence of like 64, at 64 amino acids, there are more possible sequences than there are atoms in the universe, right? Most proteins are longer than 64 amino acids. So the question is like, how can we really, it's kind of amazing actually to think about when you think about it, that, functional proteins have been found at all, right? I mean, there's been a billion years of evolution, but it's kind of remarkable that they exist at all. But then the question is, how can we even, how do we engineer proteins? How can we like search this massive space of protein sequences to find sequences that actually do what we want? So, of course, we can draw on evolution, but really, mutagenesis is the workhorse of protein engineering. So mutagenesis meaning we're just going to characterize the sequence to function relationship for variants of a protein, use that to try to understand what's going on and optimize it. So oftentimes this means doing things like deep mutational scanning or trying to develop other high throughput techniques so that you can actually go to the lab and measure the properties of a huge number of sequence variants to try to find something that improves the property over your starting sequence, or does something like what you want. If we're thinking about something like antibody engineering, right? Typical techniques there are like immunize an animal, right? So you inject your antigen into a mouse or a rabbit or a llama, and try and basically select for antibodies, because we naturally make a huge repertoire of randomized antibody sequences, and basically like biopan for something that will bind to that antigen. We can also do that in vitro with things like phage display, where you could screen like hundreds of millions of antibody sequences against that antigen to try to find anything with any appreciable binding. But oftentimes, proteins and properties are not amenable to high throughput methods, right? Like assay, characterization, especially sort of quantitative detailed characterization, is usually very low throughput. So if we're talking about something like an enzyme or anything where you have to produce individual variants, it's going to be a low throughput. So what that ends up meaning is that protein engineers spend a lot of time staring at structures, staring at sequences, trying to look at homologs to figure out like, what can I actually try? Because you have to get that, you have to choose a very restricted number of variants or sequences to test. Or just try some random mutagenesis or like exhaustively characterize all variants and just like do a lot of assays. Okay, so in this setting, what we're talking about is a situation where we have this kind of sequence variants and actual functional measurements with them. So really the challenge here is that we need to be able to generalize from small amounts of data. If you get to see a 96 well plate worth of variants, 96 variants, can we make some useful predictions then about what will happen if we make other mutations to that protein? And so conventional methods here are really difficult because things like one-hot encodings are actually unable to generalize properly in this setting. If you're using a one-hot encoding of your sequence, so that is at each position of your sequence, you encode the amino acid there into a bit vector that's 1 for the amino acid that's there. If you use a featurization like that of a protein sequence, no model that you train on that basically directly can make predictions about a site that isn't included in your data, or can make predictions about an amino acid that isn't included in your data. So that is a big challenge, right? It's inherently limited in its generalization. So the question is, how can we leverage evolutionary priors to learn these sequence function relationships more efficiently? So basically, really what we want to rely on here is transfer learning. So transfer learning is basically a machine learning paradigm where we want to be able to take knowledge that we've learned on one problem and apply it to a different problem. So this is a great example of that, right? We can have, let's take protein language models. This is knowledge that we've learned on this problem of modeling natural protein sequences. Can we take that knowledge and apply it now to actually learning specific sequence to function relationships? So basically the idea here is, let's take protein language model. Let's use it to actually featureize the sequences. We'll put that into use those features then as the features of the sequences for doing something like Gaussian process regression, and see if we can actually predict these specific phenotypes accurately. And it turns out that if we try something like this, we're actually able to much better predict the activity or like the specific properties of a protein sequence that we haven't seen than if we were to use something like one-hot encodings or other featurizations of the sequence. And also we are able to predict it better than like evolutionary priors alone. So we can then combine this idea with also thinking about how can we search the sequence space in a more information theoretically optimal way. So, something that's really common would be to say, okay, I'm going to test some variants in the lab. I'll find the best one. I'll take that variant. I'll make some more mutations to that, and I'll go and I'll test those. And then I'll take the best one and I'll make some more mutations to that, and I'll go and test those. So that's an optimization procedure. It's basically hill climbing. So it's basically hill climbing. We're here. We're going to try some things around it. We'll take the best one and we'll go in that direction, and then we'll just repeat that process until we reach some local optimum. When everything you test is not better, then you either give up or you explore more broadly, or you've reached some local optimum and you're done. But it's very easy for that to actually miss the global optimum, which is what we would really like to find. And so if we are thinking about being able to do some kind of Bayesian prediction where we actually have an uncertainty aware models, which is really useful in a low data regime as well, then we can think about a paradigm like Bayesian optimization, which is a global optimization procedure where the idea is essentially we want to choose features or like sequences in this case to test that are trading off exploration and exploitation. So let's say we were interested in maximizing our target, like maximizing activity. Well, then the sequence that we should test has both to do with what is our mean prediction, like what do we think the activity is given our data so far, but also how confident are we in that prediction? Because if we're not confident, then there's a chance that this will actually be a good data point. And so we should explore, right? We should learn, we should test sequences that we don't have a lot of information about, because we are able to better learn the sequence function relationship and figure out whether actually that's a fruitful region of sequence space to pursue for finding an optimized sequence. Okay, so we actually put this together in a project that we worked on with MIT Lincoln Lab. So what we were doing is we wanted to design some anti-COVID antibodies. And so we started out with, we had a antibody sequence that had been identified by phage display, but it wasn't a very good binder. We generated some training data by doing mutagenesis of this protein, actually measuring the binding affinities of those antibody sequence variants to the coronavirus spike. We then used that to actually train some Bayesian sequence to function predictors, in this case, sequence to affinity. And then based on those, identified sequence variants that we thought were most likely to improve over our parent sequence. Built a library of those and actually went back and tested that in the lab. And we also tested a conventional sort of directed mutagenesis library based on this initial library in parallel. I believe this is still this might be the only work where anyone has actually really done a head-to-head between sort of a conventional design approach based on re-randomizing top variants found in this initial library with an ML approach. So we actually published this earlier this year. If you're interested in reading more, I encourage you to go check out the paper. There's a lot more information in the paper about sort of exploring different model settings and things like that I will get into here. So right, as I mentioned, we started with this initial random mutagenesis to build a training data set. And something that's really interesting that you can see here is basically, when you do random mutagenesis of a protein, most variants are breaking. So in this case, our seed antibody affinity was here around 100 nanomolar. Only about 8% of variants had 100 nanomolar or better affinities to our target, and less than 1%, like less than actually 0.1%, had strong binding affinities to our target, right? The vast majority of variants completely disrupted binding affinity. We then took this, we actually trained sequence to affinity prediction models, and then we designed our library and we tested it. And so when we actually compared our ML designed library with the conventional library that we did in parallel, where we just took the top variants, took the variants in the top sequences from that initial random mutagenesis library and re-randomized them. What we found was that actually our ML design library worked really well. We our library had, the best sequences were about 20x more potent binders than the best sequences we found with the conventional library. We improved more than 100x over the parent sequence. About 70% of our designs ended up being sub-nanomolar binders, which was quite remarkable. Beyond that, actually, the models generalized surprisingly well. So something that you tend to see in this kind of conventional mutagenesis process is as you make mutations, sort of simultaneous mutations to a protein, there's some cumulative effect, right? So if you make one mutation, they mostly do nothing maybe or lean towards breaking, but there are some that maybe have some functional improvement. If you make two mutations, you might get a slightly higher functional improvement when you just happen to have sort of two good mutations together. But as you accumulate more and more mutations, you tend to get this effect where on average, those sequences get worse, and eventually you don't even have outliers that are good. Just because the probability that you included a variant that's breaking or some set of variants that are breaking becomes very high. So as you add more and more mutations, typically you get this kind of effect where eventually you have so many mutations and everything breaks. But actually, when we looked at the ML design library, we didn't limit our algorithm in terms of how many mutations it made. We basically just said, let's find whatever sequences we think are, according to the model, have the highest probability of achieving our design objective. And so the algorithm made many simultaneous mutations. It actually ended up going to much higher diversity sequences than we had in our conventional library. But remarkably, even these sequences with 12 simultaneous mutations, and actually if you look in the paper, we look even beyond this, ended up being strong binders. So these are at the single-digit nanomolar to sub-nanomolar binding level, even though they have 12 simultaneous mutations. So the model actually was able to generalize quite well to new sequence variants. Okay, so just to wrap up, basically we can combine transfer learning and Bayesian optimization to identify high affinity specific binders. Transfer learning with large protein language models actually allows us to learn these sequence to function prediction models with limited data because we can actually draw on the evolutionary priors. And we are even able to predict sort of the function of high order variants, which might appear to be out of distribution. I forgot to mention that in our random mutagenesis library, we only included up to triple mutants. So these are well beyond sort of the training distribution. Okay, so, and then to just sort of wrap up overall, I think what I've tried to motivate is that protein language models are very powerful and useful. They learn structural and functional constraints just by looking at natural protein sequences. Our evolutionary transformer POET lets us take this further. It's parameter efficient, it's easily specializable, and it's actually a generative model of whole protein families, which allowed us to improve the state-of-the-art invariant effect prediction. Transfer learning with protein language models also then allows us to learn these sequence function models very efficiently, which we can apply to designing antibodies, but actually really in principle, you can apply this to any problem where you're measuring properties in the lab. And some future directions I think are really interesting. So one is integrating structure modeling actually with protein language models, right? So currently structure modeling is sort of viewed as a downstream problem where once you have your protein language model, then you can use it to do structure prediction. But we could imagine finding ways to really bring those models together in a better way, right? Viewing structure maybe as actually a latent variable in a generative model of a protein sequence. Understanding structural ensembles and structures of point mutants. This is a big limitation right now. Not just making point predictions of a single structure, but can we actually understand the conformational landscape of a protein? Also structures of point mutants. One of the disadvantages of evolutionary models is they're generally not very sensitive to single mutations. So how can we sort of better understand what will happen in a protein, better predict what will happen in a protein structure with single mutations? And also I think thinking about combining these kinds of ML-driven protein optimization techniques actually with physics-based approaches, right? So really the other very popular method of protein engineering, which I didn't really discuss, is physics-based approaches where you would run, say, molecular dynamic simulations or coarse-grained physics models in order to be able to actually make biophysical estimates of what will happen if you make mutations in a protein, assuming that you can model that structure well. But these really aren't mutually exclusive approaches, right? They're actually quite complementary, and I think it's would be interesting to explore some of those techniques more in the future. Okay, so that's it. With that, I need to thank lots of my collaborators who worked on these things at MIT and MIT Lincoln Lab. Also Tim, who is at openprotein.ai, and Mike, who's also at openprotein.ai, who contributed to Poet. I'll plug this again if you're interested in checking out Poet and some of these other kinds of sequence to function learning and protein design tools. Check out openprotein.ai. I'm also have my group at New York Structural Biology Center, where we work much more on protein structures and cryo-EM and things like that. So feel free to look us up if that's something that you're interested in the future. Okay, thanks. I think hopefully there's a little bit of time for questions.
Yeah, thank you very much. This is really great. This is really the state of the art. The protein language model covers many things. This is really amazing and exciting. All right, so any questions from the audience? Maybe I can start. Starting from very technical ones. In this Poet, so you condition on the many sequences but the position I is depending on the earlier part, like I-1 and the earlier part, right? So why do you choose that option? You mentioned a couple of options like a masking and a bidirectional, but you choose the probably most simple one.
We chose to make this an autoregressive model because we wanted to be able to sample from it. We wanted to be able to generate sample efficiently. That was one key consideration, and the other is we wanted to actually be able to calculate an exact likelihood. So the problem with this kind of mass language modeling approach is you can't actually calculate the probability of the sequence with this model properly. You can do like a hacky kind of pseudo likelihood where you say okay, well, I'll mask each position and I'll calculate the probability of each one, and then I'll sort of just multiply those together. But that's not actually the probability of the sequence. That's a weird pseudo likelihood that's stapling together all those marginal probabilities, or all those conditional probabilities, but it's conditioned on the wrong way, right? So that was the reason we went with the autoregressive models. We really wanted it to be a proper generative model of the sequences that we could sample from. And also because we wanted it to be able to do insertions and deletions. And so to do that, you also need it to be autoregressive, because a mass protein language model, it's fixed length. If you delete a site, it's not clear how you should really properly then calculate the score of that. And it would definitely be difficult to generate those. So that's why we went with the structure.
Okay, yeah, thank you. Any other question from other audience?
So let me ask a kind of maybe another technical question, but about the antibody part.
Yeah, sure.
Is there any link from the antigen sequence to the variable fragment sequence that you have? Is there any matching that you guys find in there?
Right. So good question. Okay, so the way we framed this was basically to not use any information about the antigen other than the affinity measurements that we got. So the only way we know anything about the antigen in this is through the experimental measurements. We didn't model it in any other way. We were basically just saying we have these functional measurements, we have the antibody sequences. We're just going to learn that antibody sequence to binding affinity to the target, and that's how we. I think we started thinking about how we could generalize that more into also including sequence information about the target. In this case, we weren't changing the target, so it would have been a constant anyway. But I think, there are definitely some other problems where that would be very interesting. So actually we did look a little bit at like anti-HIV antibodies, where there's actually data for antibody sequence variants and HIV variants. And of course, now for coronavirus, this would also be very interesting because there's all the spike protein variants and like escape mutations and things that people are concerned by. But no, we didn't look at it here. But that would be, I think that's definitely also a very interesting future direction here, is to start to try to like bring in target information as well and generalize across that axis.
Very cool. Thank you.
Mm-hmm. Okay. I see there's one question on chat box. Okay, so from Ismaila. In training, how do you deal with capturing unnatural amino acids?
That's a good question. Yeah. No, unnatural amino acids are really interesting for this, actually, right? So the problem is, because we're training on natural protein sequences, we don't have any unnatural amino acids. So the alphabet is limited only to the set of natural amino acids. And these models don't have any like physical knowledge of what an amino acid looked like. So there's actually no way for you to incorporate an unnatural amino acid into that. The only way you could do it would be, you could sort of like, you could fine tune the model. Like you could introduce it as a new token. And then if you have some data that includes that unnatural amino acid, you could try to fine tune the model then to sort of understand it. But in lieu of that, the model has no way to do any, to make it have any understanding of what would happen with an unnatural amino acid, because it doesn't understand physics. It doesn't understand like the actual structure of the side chain or anything like that. I think that would be, that's a sort of another, I think, aspect of this idea of like actually trying to bring like structure into protein language models that would be really interesting, right? You could imagine not sort of featurizing the amino acids in the protein language model by like a learned embedding dictionary, and instead actually featurizing them by like their atomic structure or something, and feeding that into the protein language model. And then it might might be able to then generalize to unnatural amino acids or like other amino acid modifications. It could be, as one way to think about that. I think that would be very cool. That would be a really exciting, I think, like future direction for thinking about protein language models.
Can I have one naive question as well? Maybe, do these pre-trained models that would basically embed your sequence, do they need tuning, and how much general additional training you would need for them to be fine-tuned? This might be very, very naive, of course.
It depends on what kind of strategy that you want to take. I mean, so what we were focused on as a fine-tuning methodology is basically we're just using that model to derive features. We don't actually you don't have to necessarily change the parameters of that model, especially if you're thinking about in like a very small scale setting where you don't have very much quantitative data. Then just taking the features and then doing a very simple downstream model, like you just fit a linear model or like a Gaussian process in principle is not actually a very simple model, but it's pretty simple, generalizes well from good features. So you don't have to change the protein language model at all. But if you have a much larger data set, like actually in this project, we did have a pretty large data set here. So we actually did fine tune all the parameters of the model itself and made an ensemble of those and like looked at how that performed as well for predicting the affinities. Then you can do that where you fine tune the whole model, but that's not really practical with a small data set because it's so prone to overfitting. On the sort of generative modeling side, you could fine tune on a protein family. So you could say, have your pre-trained protein language model. You could take a family and all of those sequences and like fine tune it just to generate those sequences. But again, you have the situation where you are sort of benefiting from the general prior, but then you're actually changing the parameters of the model to be specific to your family. And so you still need a reasonable amount of data, and you need to be able to monitor that to make sure you aren't overfitting too badly and things like that. And so that was, part of what we were thinking about when we were making Poet is like, well, how would I specialize this model to like 5 sequences? Like I have 5 homologs, like there's no, you can't possibly fine tune a model on 5 homologs, right? And so which is what part of also why we're thinking about this kind of like explicit conditioning process where it's like, okay, we're going to prompt the model with those. We don't have to touch the parameters of the model, and it can sort of specialize that way. And that ended up, working really nicely. But yeah, otherwise, but if you have a larger data set, you can also just, you can do some fine tuning there to change the actual parameters of the model. And it can work quite well.
Thank you.
Mm-hmm.
Okay, there's another quick question on the chat box. So how do you deal with post-translational modifications?
Yeah, I mean, same problem as unnatural amino acids, right? As a sort of natural sequence model, it doesn't have any information about post-translational modifications. Now, that said, it doesn't have any information about them, of course, in the input features. But that said, there is natural selection on those, right? Because those are oftentimes important for the protein to function, or they cause a fitness cost if there's like post-translational modifications that are made to the protein that then disrupt its function because of introducing some amino acid into it that then is modified. And so as a result of that, there is definitely some information in the model relating to like, will this be post-translationally modified, and like, is that relevant for predicting fitness, and therefore it has to be something the model can understand, but it has to but it's purely based on that sequence context. So that would be the way. I mean, yeah. So what I think it means you're likely to see is that it will push away from modifications or like from amino acid substitutions or sequences that would be negatively post-translationally modified, and therefore would cause that to not be a fit sequence, because we wouldn't see that in like a natural family then, or we would be less likely to see it.
Okay. I have maybe last quick question. So I think the poet maybe also already, you mentioned about the structure. So probably, poet does better than ESM4 for if you just attach the structure module, because poet understands.
That's on my to-do list.
Right. Oh, okay.
That's on my to-do list.
Okay. I thought you have already done that then.
Okay, so we have looked at secondary structure prediction, and it is more accurate for secondary structure prediction by a pretty large margin, actually. But we have not done a complete structure prediction training yet. Yeah, it's on the to-do list, but we haven't done it yet.
Sure.
Yeah.
Okay. So if there's no other questions, let's thank to Tristan again. That was really, really fantastic talk. Thank you very much for all this preparation, and I really appreciate, you started from very beginning, going to the really and the state of the art, the application. This is fantastic talk.
Yeah, thanks.
Thank you.
Thanks for inviting me. It was fun.
Thanks so much, Tristan. Good to meet you.
All right. Thank you very much.
Thank you. Okay,
Thank you.
bye.
Yeah, bye. Thank you.
Thank you.